Okay, so David, you see, I have revisited also the title because it was too long and probably too multidisciplinary and too complicated for a 20-25 minutes talk. So I changed to this shorter title, and the aim of this talk is just to revisit all the concepts in both from with some of them specific, some of them shared between the two domains of toxinology, and to speak a little bit about novel concepts that I think should be also incorporating in venom research, particularly in clinical toxinology. So here are both domains of toxinology in this slide. You see we have here ecological toxinology, where the main purpose of venom is the prey subjugation, not necessarily killing the prey, but prey subjugation, and it is driven by natural selection. And on the other side, we have the clinical toxinology, which is anthropocentric biased, and has nothing to do with the type of prey that the snake that produced the venom has its preferred type. And they have also something in common, which is technology. We have several aspects of venom research, like general venomics, but also ecological venomics, translational venomics, etc. And as you mentioned, antibiomics, and all of them are based on omic technologies, genomic, transcriptomic, proteomics. And you see here there is this little mouse, which is also a link between both domains, particularly to translate figures and parameters that are gathered from this domain to the toxinology domain. I will put several examples on this. So one of the concepts that I think should be revisited is the LD50. This is a concept that was developed in the 30s, and is that simply provides a generalized indicator of the toxicity of the substance that is being tested over, and for this you need a certain period of time of exposure to determine the different doses to determine this figure. However, as we all know, snakes that have different toxicity, venoms with different toxicity, are difficult to compare, because you need to have other parameters in the equation, like the preferred prey type. Do they have the same preferred type? What is the venom yield of each of these snakes? Are there competition with the sympatric species, etc.? If there is no competition, the comparison of venoms in the same context is almost not useful. And we are also very well aware that even generalist species, like those here, like the first part of this graphic, they have quite different toxicity towards different prey. Most of them have as preferred prey, or the prey that is more affected by the toxicity of their venoms, mammals like this, rodent species, but also other lizards, and none of these has preferred type prey anurans, and you see here that the toxicity is quite different. So this means that comparing different toxicities in different species, in different contexts, is difficult. But what is the sense of the LD50? The LD50 is, as we saw, the median lethal toxicity. But is this the ecological parameters that has evolved in nature? Actually, if we see this series of graphics, the main purpose of venom usually is not killing, it's just immobilizing, subjugating the prey by immobilization, so that the snake can handle the prey without the risk of breaking its neck in the struggle. If you immobilize a prey rapidly, you have more advantages that if the prey still can struggle in your mouth. So this is one thing that should be taken always into account, but that when comparing LD50s, even from different snakes against the same prey type. And you see here, this Athropoides picadoi needs 4.4 LD50s for immobilizing the same prey, but Crotalus simus needs 6 LD50s, and these two need 2.5 LD50. So here, who is more effective in nature? Difficult to say, depends on the context of the ecological setting. And one example is this nice study that I will show on this nice Rhinobotrium bovali species, the Costa Rican false coral snake, which has a very simple venom. You see just seven main components, and only from 3 different toxin families. This venom is absolutely non-lethal for mice. You see here the animals were injected intracranially with 900 micrograms of venom per mouse, and they are still as happy as if they had not received any dose. And this is 2.5 hours later. However, as we see here, they are very effective in mobilizing iguana, which is the preferred prey. And what is curious is that even if the prey is absolutely immobilized, it is alive. Look here, let me see. Here, it is breathing. And here you will see also the iguana, the immobilized iguana breathing, but in a different manner, here with the lungs and here with the trough. And so this means that different toxins have also different immobilization effects on the same prey. So it is clear from this example that like in other taxa in nature, they immobilize and use the live prey for their own for their own purposes. Here, like spiders that pack their lunch into skill for eating the live prey, or these wasps that turn the cockroach into zombies, and after laying the eggs here, when the offspring emerge, has already had its lunch, and this is the main point. So there is a convergence in immobilization rather than killing the prey in some venoms that need to be into consideration when you try to make sense of a simple parameter like the LD50. And here you see also that with different amount of venom that deployed in the iguana, the time to immobilize decreases also. So here, then this means that we need to change perhaps the definition of the ecological parameter from the LD50 to the minimal amount of venom that immobilizes the prey in the shortest time. And this brings me also to another parameter that is so common in science in general, that one doesn't all is not people do not think much about how to determine it, which is the amount. The amount can be determined by weighting a substance, but also by comparing certain parameters like a color in a standard curve constructed with a standard protein, but the standard protein is always a good standard for this protein, but in many times it's not a good standard for other proteins. And this is the case for many venoms, particularly when the venoms are composed by small toxins that do not have the same colorimetric reaction as the standard that is being used for the standard curve. So what we are proposing and we are using in the lab since some time is to determine the extinction coefficient, which is a parameter that is a parameter from the substance that you are analyzing. And you can do this in whole venoms just by weighting a certain amount in a precision balance, dissolving it also by weight in the same way, calculating the milligrams per ml that you have here in this solution, determining the absorbance at 280 and dividing absorbance by the concentration, and then you have the extinction coefficient 0.1%. This is the absorbance of a 1 milligram per milliliter at 280 nanometers in a 1 centimeter cubic of path length, light path length. There is also another method that works pretty fine, which is to calculate the same extinction coefficient for this venom, but taking into consideration the chemical structure of the components of the venom. Here, these are the major components of this venom. These are the molecular masses. These are the number. This is the number of tryptophan, tyrosines, and cysteines. And with this formula, you can estimate an extinction coefficient which is almost identical as the experimental one. And the same happens for this other a little bit more complicated venom, where you get the same result in both manners. So this is now very useful, because once you have determined the extinction coefficient in one way or another for a venom, you can use it always without necessity to determine it again. You just need to determine the absorbance of the solution that you are using, for instance, for determining the LD50 or ED50s or whatever parameter you want to measure with this venom. So this is very convenient. So now let's go to how to make sense from an LD50 and accurately determine LD50 in model organisms into clinical toxinology. Here it's clear that you cannot just translate linearly this figure from the animal to human. You need something that is called allometric scaling. This is based in the fact that many observations since the beginning of the last century, that there is a linear relationship between the metabolic rate and the body mass, not only of different range of life, in both in unicellular, in multicellular, ectotherms, endotherms. There is a linear relationships that adjust to a power function. So in different many people have studied this relationship and have come out with a formula for scaling from data that have been gathered in an animal to human. And in the case that for translating an LD50, for instance, that has been gathered in mice to human, is very simple. You just need to take the animal dose in milligrams per kilogram, per instance, and multiply by the ratio between the animal km, that is the kilogram in square meters of body surface area. This is the metabolic parameter from animal divided by human. So this means you multiply the LD50 gathered in mice by 3 and divided by 3 and divided by 37, or just divided by 12.33. And then you get an accurate an insight on what should be the toxicity for human. And this is important because, as you see in this slide, this is X ocellatus, the LD50 of 1.4 microgram of venom per gram of mouse body weight is 28 micrograms for a 20-gram animal. If you translate this linearly, this means that you would need 1.4 times 6,000 grams of body weight. This means 84 milligrams of venom for killing half population of humans of 60 kilograms. And this is an absurd, simply because we know that X cause death, and the average venom yield of different X species, particularly of this X ocellatus here in Ghana, is 11.2 milligrams. So if you need 84 milligrams to kill a human of 60 kilograms, you would never observe a death caused by X ocellatus here. And the same happens to any other species that you want to apply this. So using the formula, you can easily calculate that actually the LD50 for human is around 6.8 milligrams per individual. So this means you have between almost 2 to 4 or 5 LD50s in this venom, in these venoms. Of course, it is important also something that is rarely measured, which is the amount of venom that is deployed in a defensive bite, and not in a forced experiment where you press the glands and obtain the maximum of venom that is that you can extract from the glands. But what we need in the clinic to know is what's the venom that a species will deploy in a defensive bite, which is always what causes a human accident, not a predatory bite. And here is let me just. Different snakes have different habits of biting, and of course different yields of venoms. Important, although rarely done in venom research, is also to determine the toxicity of the individual components, because the components is the ones that you need to neutralize with proper antibodies to reverse the effects of an envenoming. So not only the LD50 for the whole venom, but also how should you let's say mix two sets of antivenoms, one against the neurotoxins, one against the cytotoxins, to neutralize the effects of a venom that contains both components. Here we have different. It's like trying to solve in this picture is just trying to solve a single equation, one equation with many different unknowns. Here we have the relative abundance. We have here the LD50 in mice, and they translated to human LD50 for the different components. And how can we get here can make sense of this data? Well, sometimes here also the ecology and venom variation can help us. And this is a good example. Here we found the same. This is the same species but it's a variant, a geographic variant of the venom. It's a venom that does not contain almost neurotoxins. We have also the same, exactly the same chemical cytotoxins in these proportions. So if we can estimate here that the whole venom contains 35 human LD50s for this average venom yield, and we determine the LD50 for this other variant, which is 0.28 milligrams per kilogram of human, which amounts to 18.6 human LD50s. The difference between both is the contribution of the neurotoxin. And here now we can calculate that 46% of the lethality of this venom is due to the neurotoxins and 53.2 is due to the cytotoxins, even if they have an LD50 that are much larger than the one for the neurotoxin. So now we know how to mix different antibodies to end up blocking the lethal effects of both types of toxins. Another thing that usually the manufacturers never have in consideration is the amount of active ingredient, immunoglobulin or Fab fragments, and how these active molecules interact or block the venoms that are intended to be neutralized. Here in this antivenomic analysis, what is in gray is the data for mouse, and you see that for the maximum LD50 neutralizing potential per vial for this antivenom is 116.5 mice LD50s. If you translate this for human, you end up with only 0.45 LD50s for human. And look at here also the different LD50s for human, also for mice, of course, for human, for the different venoms that the antivenom is intended to neutralize. You have from 2 LD50s that need to be neutralized to almost 600 something LD50s for some species like this Naya AG from Uganda. So the thing is that if you take the insert in the package, what is specified as potency for many antivenoms is about 250 LD50 per vial, and this 250 translate only in one for each of this venom, in one out of 18, 4, 60, 28, 34, etc. So they have been normalized for a certain amount of mice LD50, but without having into account the different venom yields and the different amount of LD50 for human that the species, the target species, can deploy in the victim. So this is a nonsense of at least from the point of view of the design of an antivenom, and this should be changed in the future. Another thing that needs also to be changed is how to express the neutralization capacity of an antivenom. Usually this expressed is as an ER50, which is the milliliters of antivenom that is needed per a number to neutralize a number of LD50s. However, what is needed is not relate to the LD50, but to to saving the whole population, not only half of it. So the potency which takes.
Sorry, Juan, 5 minutes to go, Juan. Sorry to
yes,
interrupt.
this is my the next one is the last one, and so here potency takes into account the neutralization of all the LD50s that have been deployed in the venomic, not only all the LD50 minus one that is not neutralized at the end of the assay. And this is probably this is expressed in this way because the ER50 depends on the number of LD50s that have been used to calculate the ED50 of the antivenom. You see here for smaller number of LD50, it can be as much as 2 times the real potency of the antivenom. And only when you increase sufficiently the number of LD50s used in the assay, they convert to the same figure. And the potency doesn't depend on the number of LD50s that has been employed. So usually what is in the insert that is related to this parameter is about 1.5 to 1.6 higher than the real neutralization potency of the antivenom. So this should be also taken into consideration by the regulatory agencies. So just to conclude, my impression is that, among others, antivenom should be designed for the context of human envenomings, and the aim should be to save human, not mice. Antivenom production should be based on neutralizing strategic toxins, not necessarily the whole amount of venom. The neutralization capacity of an antivenom should be expressed as the potency, as I commented in the last slide. The potency of the antivenom should be, of course, asymmetrically adjusted to neutralize the disparate lethality of the target venoms, not the same number of LD50s for mice. And the antivenom package insert should also include basic but very useful for testing the antivenoms information, such as the amount of active molecules, the immunoglobulins or part of them in the vial. So these are just some thoughts that I thought were interesting to share between a multidisciplinary audience like this. And with this, I thank you for your attention and am ready and open for any question that the audience like to ask me. Thank you.